Good morning. I am Dr. JAYAKALA. Today we will be having discussion on sulfur containing amino acids. The sulfur containing amino acids are methionine, cysteine and cysteine. So the methionine, if you see the structure of methionine, it has the methyl group attached to the sulfur atoms. Whereas cysteine, it has the sulfidyl group. This is the special group present in the cysteine. Cysteine is formed from two molecules of cysteine on oxidation. So the sulfidyl group will be releases the H plus ions. In cysteine, the disulfide SS bond will be present. So this is the special feature of the sulfur containing amino acids. Methionine is having methyl group, special methyl group attached to the sulfur atom, whereas cysteine is having sulfidyl group and cysteine will be having disulfide bond. Methionine is sulfur containing essential and glucogenic amino acid. Essential, you know that it is not synthesized in the body, it is only supplied through diet. And glucogenic amino acid is nothing but the product of methionine, metabolic product of methionine enters in the synthesis of glucose. That is the pathway in the enters in the gluconeogenetic pathway in the synthesis of glucose. So, methionine is the glucogenic amino acids. Methionine is abbreviated as three-letter word met and a single letter word M. The degradation of methionine results in the synthesis of cysteine. So cysteine is synthesized from the methionine. So cysteine is not a it is a non-essential sulfur containing amino acid. The metabolism of sulfur containing amino acids are categorized into or grouped into three pathways. One is the activation of methionine and transmethylation reactions. Second will be conversion of methionine to cysteine. And third will be degradation of cysteine. Under three categories, we'll see the metabolism of the sulfur containing amino acids. The first pathway will be the activation of methionine and transmethylation reactions. So the activation of methionine to SAM, that is S-adenosyl methionine. Why the methionine is first activated? Because as I told, it has the methyl group in its structure. So it is the source of methyl group in our body, methionine. But methionine is not easily releases the methyl group, cannot easily release the methyl group. So for that, it has to be activated first. So the methionine should be activated to S-adenosyl methionine. This activation is done by the enzyme methionine adenosyl transferase, that is MAT. Here what happens, the adenosyl group from ATP is attached to the sulfur atom. So the sulfur atom becomes the sulfonium ions. So it can easily donate the methyl group. So the methionine is activated to active methionine, otherwise called S-adenosyl methionine. The enzyme is methionine adenosyl transferase. See, methionine, you see the structure of methionine, the bond between this CH2, sulfur and CH3 is very stable bond. It cannot break the bond and releases the methyl group. So once it gets activated by addition of this adenosyl group to the sulfur, so the sulfur becomes the sulfonium ions and there is a high energy compound. So, which is denoted by squiggle bond. The high energy compound is denoted by the bond squiggle bond. So now the methyl group is the bond between the methyl group and sulfonium ions is very labile, very weak bond. So it can easily releases the methyl group from its structure. So this activated methionine is called S-adenosyl methionine. The reason for activation is for the release of methyl group from the methionine. The enzyme is methionine adenosyl transferase because there are three isoforms of MAT, abbreviated as MAT. 1, 2, and 3. 1 and 3 are present only in liver, whereas 2 will be present almost in all our cells. And SAM is nothing but S-adenosyl methionine. It is the main source of methyl groups in the body. So how the methyl group is released or donated in methionine, the thioether linkage. This linkage is called thioether linkage. That is CH2 sulfur and CH3. That linkage is thioether linkage. That is very stable. But in SAM, that is S-adenosyl methionine, due to the presence of high energy bond, the methyl group is very labile, very weak, and it can be easily transferred to the acceptors. So methionine is activated to S-adenosyl methionine, and the enzyme is methionine adenosyl transferase. Now the next step will be the release of the methyl group from S-adenosyl methionine. Once the methyl group is released or donated, the acceptor accepts this methyl group. So the acceptor becomes the methylated acceptor. Now, once it releases a methyl group, it becomes the S-adenosyl homocysteine. S-adenosyl methionine becomes the S-adenosyl homocysteine after removal of the methyl group. So, the methyl group is accepted by the acceptor. So, if you see the difference in S-adenosyl methionine and S-adenosyl homocysteine, just the methyl group. Presence of methyl group is S-adenosyl methionine, and the methyl group is removed, it becomes the S-adenosyl homocysteine. Here the enzyme is methyl transferase. Enzyme is methyl transferase, and the acceptor becomes the methylated acceptor, and SAM becomes S-adenosyl homocysteine. Now the third reaction is the adenosyl group is released from the S-adenosyl homocysteine. It is homocysteinase or adenosinase. Homocysteinase enzyme brings about the removal of the adenosyl group from S-adenosyl homocysteine. Now, what is formed? Homocysteine. Once the adenosyl group is removed, it becomes the homocysteine. Homocysteine is the homology of cysteine. So, cysteine is synthesized from homocysteine. If you see the difference, structurally, the difference between methionine and the homocysteine is only the presence of methyl group. Demethylated methionine is the homocysteine. So, from homocysteine, methionine is formed by remethylation. So, the methyl group is originally um, originated from the methyl cobalamin. So the methyl group is the source of methyl group for methionine is the one carbon pool, that is the methyl cobalamin. The methyl cobalamin transfers the methyl group to methylene tetrahydrofolate, and the methylene tetrahydrofolate is reduced to methyl tetrahydrofolate, and the methyl group is donated to homocysteine, and the homocysteine is converted to methionine. So this is the cycle. Methionine to S-adenosyl methionine, and S-adenosyl methionine to S-adenosyl homocysteine, and S-adenosyl homocysteine to homocysteine, and homocysteine to methionine. Here the enzyme is homocysteine methyl transferase that brings the addition of methyl group to homocysteine, and homocysteine is methylated to methionine. From homocysteine, the cysteine is synthesized. So what is the second category? Metabolism, the conversion of methionine to cysteine. So we'll see how cysteine is formed from methionine. So it's from methionine, homocysteine is formed. From homocysteine, cysteine is formed. So homocysteine is uh, the S-adenosyl homocysteine As I told, the adenosine group is released and it forms the homocysteine, which is the higher homologue of cysteine. From homocysteine, methionine is synthesized, that is methylated, remethylated to form methionine. So that enzyme is called homocysteine methyl transferase. The methyl group is originated from B12, that is the one carbon pool. From how the homocysteine is degraded and form the cysteine, that is the second category of metabolism of sulfur containing amino acids. First, homocysteine is condenses with the serine to form cystathionine. So this is catalyzed by the enzyme cystathionine beta synthase. So this enzyme is dependent on the pyridoxal phosphate, that is the coenzyme of B6. PLP is the coenzyme for cystathionine beta synthase. I'll show you the pathway. Homocysteine, it combines with the serine, forming cystathionine. Serine is the another amino acid, that is the hydroxyl containing amino acid, hydroxyl group containing amino acid. So homocysteine condenses with the serine, forming cystathionine. The enzyme is cystathionine beta synthase. The coenzyme is pyridoxal phosphate. Cystathionine, the next will be degradation will be cystathionine hydrolyzes to form homoserine and cysteine. So now the cysteine is formed from homocysteine and homocysteine is formed from methionine. So if you see, the cysteine is formed from the methionine. So if you see the structure of cysteine carefully, it is nothing but the carbon and hydrogen skeleton is completely from the contributed from serine, the amino acid serine, and the sulfhydryl group is originated from the methionine. So this sulfur group is originated from methionine and the carbon hydrogen skeleton from serine. This transfer of sulfhydryl group from methionine to cysteine, it is called transsulfuration reaction. It is called transsulfuration reactions. So the structure is completely the serine and the sulfhydryl group transferred from methionine. Just transfer of the sulfhydryl group. So transsulfuration reactions just brings the sulfhydryl group from methionine to serine and forming the cysteine. So this enzyme is hydrolyzing enzyme of cystathionine is called cystathionase. Cystathionase. After cysteine is formed, it is degraded, that is deaminated, that is removal of ammonia, and decarboxylated, the removal of carbon dioxide. Finally, it forms the propionyl CoA. So propionyl CoA is the odd chain fatty acid. It forms from the odd chain oxidation of odd chain fatty acid. The end product is propionyl CoA. The propionyl CoA enters the gluconeogenetic pathway. So this is one of the important precursor or source of gluconeogenesis. How the propionyl CoA is converted to glucose. The propionyl CoA is first metabolized to L-methyl-malonyl-CoA and then it is isomerized to D-methyl-malonyl-CoA and then D-methyl-malonyl-CoA is mutase to succinyl-CoA and the succinyl-CoA enters the TCA cycle. From that the gluconeogenesis pathway occurs. Finally the glucose is formed. So cysteine is also a glucogenic amino acids, whereas methionine, that is the end product, finally it forms the propionyl CoA. So that is also a glucogenic amino acid. But cysteine is the non-essential amino acid because cysteine will be produced from the methionine, whereas methionine is essential amino acid. Only it can supply through diet. So it's each reaction is mentioned here. Homocysteine combines with the serine forming cystathionine. The enzyme is cystathionine beta synthase. PLP, pyridoxal phosphate, is the coenzyme. Next step, cystathionine hydrolyzed to form homoserine and cysteine. cysteine. So cysteine is completely contributed from serine and sulfidyl group from methionine by transsulfuration reactions. The cysteine now deaminated and decarboxylated to form propionyl-CoA. The propionyl-CoA is the important precursor for the synthesis of glucose. So cysteine and methionine is the glucogenic amino acids. So this is the final oxidation, deamination and decarboxylation to form propionyl-CoA. So why the methionine is activated and how it donates the methyl group we have seen. Why the methionine is activated? Because it has to donate the methyl group. So here we are giving the list of methyl acceptor and the methylated product. This reaction is called transmethylation reactions. So the substrate guanidoacetic acid. Guanidoacetic acid is the combination of condensation of arginine and glycine. This guanidoacetic acid, when methylated, that is addition of the methyl group methylated from methionine, that is from active methionine, it forms the creatine. Creatine is the high energy compound. Guanidoacetic acid is methylated to form creatine, and norepinephrine is methylated to form epinephrine. Very important. Catecholamines, epinephrine. And epinephrine is methylated to form metanephrine. And ethanolamine, it is methylated to form choline. Very important for the conduction of nerve impulses, choline. And again, lysine, methylated lysine is called methyl lysine. It is given, that is another name for methyl lysine is carnitine. Carnitine is very important long chain fatty acid transporter. It's very important for beta oxidation. And methylated tRNA, methylated DNA is very important for the translation and transcription. So now out of which we'll see important methylated products. One is creatine, the other will be epinephrine, choline and melatonin. We'll see one by one. Creatine. Creatine is the methylated guanidoacetate is to form creatine. The methylated guanidoacetate is finally forming the creatine. The creatine is stored as high energy group, high energy compound in the muscle as creatine phosphate. So the creatine phosphate is stored in muscle as high energy compound. So it is a very important methylated product. Next will be epinephrine. The methylated norepinephrine is methylated to form epinephrine. Important catecholamines. Epinephrine is very important for the maintenance of the blood pressure, and it is very important for the myocardial contractility. So epinephrine is almost most of the time it is a many times it is a life-saving drugs. So it can save the life of a person. So it's a life-saving drug. Epinephrine, it is methylated norepinephrine is forming the epinephrine. From epinephrine, the metanephrine is formed by the enzyme catechol O-methyl transferase, otherwise simply abbreviated as COMT. This enzyme has the clinical application because the inhibitor of this COMT is used in the treatment of Parkinsonism. And also the COMT is very important. The inhibitor of COMT keeps the dopamine in a high level in our body. So it's very important enzyme, inhibitor of the COMT. Next will be choline. It is methylation of the ethanolamine produces choline. Choline is very important for the conduction of nerve impulses in the nerve endings. And melatonin, it is methylated acetyl serotonin. And this is also very important for maintaining the rhythm in our body, like circadian rhythm. It's very important for in our body. Carnitine, it is methylated lysine. It is called carnitine. Carnitine is very important transporter of lipids. So in beta oxidation, the long chain fatty acid from cytoplasm are transported to mitochondria by carnitine transporter where in the mitochondria the beta oxidation occurs. So the long chain fatty acids cannot permeate into the mitochondria. So this carnitine acts as a transporter and transports the long chain fatty acid from cytoplasm to mitochondria for oxidation, beta oxidation. And DNA methylation and tRNA methylation are important for transcription as well as the translation. This transfer of methyl group from one substrate to the from acceptor to transfer the methyl groups are called transmethylation reactions, and these are carried out with the help of the S-adenosyl methionine, otherwise called active methionine. The methyl groups are originally derived from the one carbon pole, and this that is from methyl cobalamin. The methyl groups are originally derived, and the methyl group is from methyl cobalamin is transported from methylene tetrahydrofolate to methyl tetrahydrofolate, and from methyl tetrahydrofolate it is transferred to homocysteine, and then homocysteine is converted to methionine, where the B12 is the important coenzyme. And folic acid also very important for the conversion of homocysteine to methionine. So any deficiency of folic acid or vitamin B12, the methyl group is trapped. This is explained as folate trap, where a severe methionine will not be formed and homocysteine will be accumulated. Again, homocysteine accumulation in the plasma is having a very clinical, having important clinical application. I'll explain later. Now we'll move on to the cysteine. So cysteine is having the sulfhydryl group, and it is a non-essential because it can synthesized from methionine. So it is a non-essential amino acid, and it is glucogenic amino acid because the product of cysteine, that is the propanyl CoA, enters the gluconeogenetic pathway. So it is a glucogenic amino acid. Cysteine is present in large quantity in the hair and nails because the keratin, cysteine is normally present in large quantity in keratin. So keratin is normally present in our hair and nails. So cysteine is normally present in large quantity in the hair and nails of our body. Formation of cysteine already I have explained because that is the homocysteine combines with the serine, condensed with the serine, forming the cysteine. First cystathionine and then to cysteine. So if you see the structure, the sulfur group is originated from methionine by transsulfuration reactions. If you see the overall reaction, methionine is converted to S-adenosyl methionine, and S-adenosyl methionine, the methyl group is removed, it becomes the S-adenosyl homocysteine. The adenosyl group is removed, it becomes the homocysteine, and the homocysteine is condensed with the serine forming cystathionine, and the cystathionine is hydrolyzed to form finally the cysteine. So it is briefly explained as methionine to cysteine cycle, methionine cysteine cycle. I will explain in detail. Methionine, it is when it releases the ATP and the adenosyl group is attached to a methionine, forming S-adenosyl methionine. This is the abbreviated as SAM. This is nothing but the active methionine. So the enzyme is methionine adenosyl transferase. This S-adenosyl methionine transfers the methyl group to the acceptor, and the acceptor becomes the methylated acceptor. So once the methyl group is removed, the S-adenosyl methionine becomes the S-adenosyl homocysteine. The enzyme is methyl transferase. The S-adenosyl homocysteine, when the adenosine group is removed, it becomes the homocysteine. The homocysteine remethylated to form methionine. The methyl group is originally derived from the one carbon one carbon pool, that is from the methyl tetrahydrofolate, transfers the methyl group and then to a homocysteine, and it becomes a tetrahydrofolate. The methyl group is originally derived from the methyl cobalamin. The enzyme is methionine synthase. So from homocysteine the methionine is formed. Enzyme is methionine synthase. The methyl group is originally derived from the methyl cobalamin, and the methyl tetrahydrofolate transfers the methyl group from one carbon pool to homocysteine. From homocysteine, cystathionine is formed by cystathionine enzyme, synthase enzyme, where PLP is the coenzyme, and from cystathionine the cysteine is formed. Again the B6 is the PLP is the coenzyme. So now the cysteine is formed from methionine to cysteine. It is explained as methionine cysteine cycle. How the cysteine is degraded? So degradation by transamination and degradation by decarboxylation. So transamination, cysteine is transaminated first to form beta mercaptopyruvic acid and then to pyruvate. So pyruvate is again an important product for the gluconeogenesis. From pyruvate, how the glucose is formed, it has been explained in gluconeogenesis pathway. So cysteine is first transaminated to form beta mercaptopyruvic acid and then to pyruvate. This beta mercaptopyruvate has sulfur group. That sulfur group can be transferred to cyanide group to form thiocyanate. And then decarboxylation. Cysteine is decarboxylated to form beta mercaptoethanolamine. So beta mercaptoethanolamine is the component of coenzyme A. So coenzyme A is the composition of beta mercaptoethanolamine with pantothenic acid, the B5 vitamin. So B5 pantothenic acid and beta mercaptoethanolamine forms the coenzyme A. So beta mercaptoethanolamine is formed from decarboxylation of cysteine. So cysteine is very important for the formation of synthesis of coenzyme A. Coenzyme A has many roles in our body. Synthesis of succinyl-CoA, synthesis of acetyl-CoA. So it is very important product formed from cysteine on decarboxylation reaction. So cysteine is first transaminated to form beta mercaptopyruvate, and then from beta mercaptopyruvate, pyruvate is formed. This is important precursor for gluconeogenesis. On decarboxylation, cysteine, see, one thing you should be remember that transamination reaction, pyridoxal phosphate, the B6 is the coenzyme, and all decarboxylation reaction, the B6 is the coenzyme. Pyridoxal phosphate is the coenzyme. So cysteine on decarboxylation, it forms the beta mercaptoethanolamine. The beta mercaptoethanolamine combines with the pantothenic acid, forming the coenzyme A. What are the important metabolic functions of cysteine? We'll see one by one. First will be the formation of glutathione. So glutathione is the gamma glutamyl cysteine glycine. So it is the composition of three amino acids: glutamic acid, cysteine and glycine. So it is a important, biologically important tripeptide in our body. So you should know the difference between tripeptide and trimer. Tripeptide is having the three amino acids, whereas trimer is having three polypeptide chains. So glutathione is biologically important tripeptide, is having three amino acid: glutamic acid, cysteine and glycine. Normally, glutathione is generally abbreviated as GSH because it has the cysteine amino acid in its structure. So, cysteine has the sulfidyl group, that is the reduced form of um, sulfidyl group. So, it has SH group. So, the glutathione is always mentioned as sulfidyl group along with that. So it shows that the glutathione is in a reduced form. When the hydrogen group is there, it is a reduced form. When it becomes oxidized, it becomes the disulfide group. So the glutathione is always abbreviated as GSH, indicate that the sulfidyl group, reactive sulfidyl group is present in it. It was first isolated in 1921 by Sir Frederick Hopkins, and he was awarded Nobel Prize for its isolation of glutathione. So first glutamate combines with the cysteine forming gamma glutamyl cysteine. So where ATP is utilized and it is converted to ADP and pyrophosphate. This gamma glutamyl cysteine combines with the glycine forming glutathione. So here also second ATP is utilized and which is converted to ADP and pyrophosphate. So the formation of glutamate, there are two ATPs utilized. This is explained in the Meester cycle. This Meester cycle explains how the amino acids are absorbed in our body. So all the amino acids are absorbed through the plasma membrane into the cell. So along with that amino acid, the glutamic acid and cysteine, glycine, all the amino acids are absorbed. The once the amino acid attached to the glutamic acid is released, the glutamic acid is now formed. From glutamic acid, the cysteine present in the absorbed amino acid will be combines with the glutamate. In the presence of ATP, it forms the glutamyl cysteine, and this glutamyl cysteine now combines with the glycine. In the presence of ATP, it forms the glutathione. So two ATPs are utilized in the formation of glutathione. It is explained in Meester cycle in the absorption of amino acid. So this glutathione is important for the act as a coenzyme in all the reduction reactions. So the metabolic role of JSH, when it is written, it is the reduced form of glutathione. It is mainly used for the reduction reactions. So when GSH is reduced, it is used in the reduction reactions. Once it is used, the glutathione is reduced, and the reducing equivalence will be used to reduce the molecule. So the molecule, the glutathione becomes oxidized form, and the reducing equivalence is used to reduce the substrate. So this glutathione, when it oxidizes, it becomes the oxidized glutathione. It is mentioned as GSSG, that is the disulfide bond. So when the reduced glutathione is oxidized, it becomes the disulfide bond. Sulfidyl group becomes the disulfide bond. This reducing equivalents are utilized to reduce the substrate. For example, methyl acetoacetate is reduced to fumaryl acetoacetate in the metabolism of phenylalanine, that is the aromatic amino acid. And citric acid is reduced to taurine, where glutathione is utilized. Again, the glutathione is very important maintaining the RBC membrane integrity. How it maintains the RBC membrane integrity. Glutathione is normally present in the RBC. During the genesis, synthesis of RBC, there are free radicals formed inside the RBC. This free radical will be inactivated by the reduced form of glutathione, which is present in the RBC. So the enzyme used for the inactivation of free radical is the glutathione peroxidase, which is a selenium containing enzyme. This is also present in the RBC. So this I'll explain. I'll show it in the pathway. The O2 superoxide anion and this H2O2. These are all the free radicals formed in the synthesis of during the synthesis of RBC. When it is superscript, a molecule superscripted with a dot, that shows it is a free radical. Superoxide is a free radical. But though it H2O2, that hydrogen peroxide is a free radical, but it is not superscripted. So this is the one which is not superscripted, but it is a free radical. When this superoxide anions are formed during the synthesis of RBC, it is inactivated by superoxide dismutase, converted to hydrogen peroxide. Again, it is a free radical. It will be inactivated that by the glutathione peroxidase enzyme, selenium containing glutathione peroxidase enzyme. It is present in the RBC. This glutathione peroxidase converts this H2O2, reduces this H2O2 into water molecules. So all the reduction reactions I told you, the reduced form of glutathione is utilized. So this glutathione, reduced form of glutathione is utilized by the enzyme glutathione peroxidase on reducing this hydrogen peroxide to water molecules. This reduced glutathione will be converted to oxidized glutathione, that is GSSG. So we always need a reduced form of glutathione in the RBC to maintain the membrane integrity. So what happens? This oxidized glutathione will be now converted to reduced glutathione by the enzyme glutathione reductase. How it reduces the oxidized glutathione? It utilizes the NADPH and H plus formed from HMP shunt. So this NADPH and H plus having the reducing equivalence. So this utilizes this reducing equivalence, the glutathione reductase, and reduces this oxidized glutathione into reduced glutathione. That is GSH is formed. That GSH is utilized by the enzyme glutathione peroxidase to inactivate the free radical to water molecules. Again, it becomes oxidized. The oxidized glutathione will be converted to reduced glutathione by the enzyme glutathione reductase by utilizing the NADPH and H plus. This reduced will be converted to oxide. This is a cycle. So always it's a cycle, and always in the RBC, the reduced form of glutathione will be present to inactivate the free radical formed from the during the genesis of RBC, and to keep the RBC membrane integrity, maintain the RBC membrane integrity. Suppose the reduced form of glutathione is lost, it's not available, and the membrane integrity is lost. So it leads to hemolysis. That is the release of blood, hemolysis, lysis of RBC. So first we see the maintaining the RBC membrane integrity, how the glutathione is utilized in maintaining the RBC membrane integrity. And second will be the conversion of methemoglobin to normal hemoglobin. Methemoglobin is nothing but the oxidized hemoglobin. Oxidized form of hemoglobin is the methemoglobin. Normally, if you see the structure of hemoglobin, it has the heme has the iron in its center. The iron is always in the reduced form. Why? Because once the iron is in a reduced form, it can easily carry the oxygen from lungs to and transport to the tissues. So the iron present in the heme is always in the reduced form. That reduced form is called ferrous form, ferrous state. So once it becomes, the hemoglobin becomes oxidized, the iron present in the heme becomes the oxidized form called ferric form. So that ferric form present in the hemoglobin is called methemoglobin. So the disadvantage of methemoglobin is it cannot carry the oxygen from lungs to tissues. So that is a disadvantage of methemoglobin. So the methemoglobin is converted to normal hemoglobin. That is, the iron is in the reduced form by glutathione. So glutathione, that is GSH, the reduced form of glutathione, reduces the methemoglobin to normal hemoglobin. So the glutathione will be oxidized to GSSG and the hemoglobin is reduced. So it can easily carry the oxygen from lungs to various tissues. That is the advantage of reduced hemoglobin. That is a normal hemoglobin. So glutathione is used to reduce the methemoglobin to normal hemoglobin. Next, glutathione is important for the conjugation reactions for detoxification. Detoxification is nothing but the process of converting toxic reactions material to compounds to non-toxic compounds, and that non-toxic compounds can easily excreted via kidneys. For example, any poisoning like organophosphorus compounds, halogenated compounds, nitrogenous substances, or heavy metal poisoning, or overdosage of some drugs, that is during drug metabolism, this glutathione is utilized, and that will be conjugating these compounds, and easily it is converted to non-toxic compounds. The conjugation is the final reaction occurring during the phases of detoxification, like hydroxylation, reduction, oxidation, and finally conjugation. So conjugation by glutathione is very important for the detoxification of poisoning, various compounds poisoning, metal poisoning, and also the overdosage of any drugs accidentally we take, that will be metabolized in the liver. So this conjugation occurs mainly in the liver, and the enzyme utilized for this conjugation reaction is glutathione S transferase GST, and this enzyme is present only in the liver, and conjugation occurs only in the liver. And next will be the activation of enzymes, how the enzymes are activated. The glutathione having the sulfidyl group, which is present in the active site, always keeps the enzyme in the reduced form. So this reduced form of the enzyme is always very active in its nature. So the glutathione present in the active site will always keep the enzyme very active. And next will be the formation of taurine. The cysteine is oxidized to citric acid and then to decarboxylated to form taurine. Taurine is again very important conjugating agent. It conjugates the bile acids and forming the taurocolate. Folic acid and uh, mm, folic acid is important. Bile acids, and these folic acids are conjugated by the taurine. Taurine is produced from synthesized from cysteine on oxidation and then decarboxylation forming the taurine. And taurine is important modulator of calcium fluxes and and calcium binding and movement. So taurine is a modulator of calcium fluxes, calcium binding, and movement. It is an important inhibiting neurotransmitter in the CNS. Taurine acts as a inhibiting neurotransmitter. So cysteine is oxidized to citric acid and then decarboxylated to form taurine. And cysteine is very important to maintain the structure of proteins. That is the configuration of proteins. The cysteine residue is present in the polypeptide chains, having forming the disulfide bridges, and it forms more active proteins and also maintains the structure of the protein. For example, insulin. Insulin has the cysteine residues in its structure. So it forms the disulfide bridges along the intracysteine molecules. So forms the disulfide bridges and keeps the insulin in its correct configuration. And immunoglobulins also has the disulfide bridges and keeps the immunoglobulins in correct configuration, that is correct structure. And also it makes the insulin and immunoglobulin in biologically active form. The presence of disulfide bridges makes the polypeptide chains in its position and its structure, and it makes it more active form. Example, presence of disulfide bridges in insulin and immunoglobulins. And this N-acetylcysteine, it is very important. It is nowadays used to improve the body levels of glutathione, because cysteine is important. Cysteine is having the sulfidyl group, and glutathione is formed from cysteine. So the N-acetylcysteine is used to improve the body level of glutathione. Why glutathione is important? I have explained the functions of glutathione. Very important coenzyme form in all the reduction reactions and maintain, very importantly, it maintains the RBC membrane integrity. So it is very important for our body. This N-acetylcysteine improves the body level of glutathione. So hence it is used as an adjuvant in the treatment of any injury caused by the free radicals. Glutathione and N-acetyl cysteine will be used in the therapeutic effect in chronic hepatitis B patients. In the erythrocytes, the rate of glutathione synthesis will be determined by the availability of L-cysteine. If cysteine is more, the synthesis of glutathione is also more. In erythrocytes, it's very important to inactivate the free radical formed during the synthesis of RBC. So there is a significant decline in the influx of L-cysteine in erythrocytes during the aging. So aging always reduces the cysteine formation. So once cysteine formation is decreased in the aging, so glutathione is also decreased. So glutathione is very important as I explained the functions of glutathione. So to prevent the aging, glutathione is nowadays targeted in utilizing the glutathione in anti-aging cream to prevent the many parameters formed during because of aging. So cysteine supplementations nowadays very important to maintain the glutathione during the aging process. This is the overall summary of methionine cysteine metabolism. Methionine, it is converted to S-adenosyl methionine, and this S-adenosyl methionine is important for the transmethylation reactions. These are methyl acceptors, accepts the methyl group, and creatine is formed, epinephrine is formed, choline is formed, melatonin is formed. So these are all the methylated product. Once the methyl group is removed from S-adenosyl methionine, it becomes the S-adenosyl homocysteine. From S-adenosyl homocysteine, the adenosyl group is removed, the homocysteine is formed. This homocysteine combines with the serine, forming cystathionine, and the cystathionine is hydrolyzed to form homoserine, and the cysteine is formed. From cysteine, pyruvate is formed. Pyruvate is important for the gluconeogenesis, that is a synthesis of glucose. And from cysteine, coenzyme A is formed, and from cysteine, taurine is formed, and from cysteine, glutathione, very important coenzyme is formed. And the metabolism of sulfur is very important because it is the sulfur containing amino acid. Finally, how the sulfur is metabolized in the body. The sulfur present in the body may be either organic sulfur or inorganic sulfur. The organic sulfur is a component of proteins from the sulfur containing amino acids, or the organic sulfur may be as a part of sulfatides, or the organic sulfur may be the part of glycosaminoglycans. Whereas the inorganic sulfur is derived from the only by the sulfur containing amino acids by transsulfuration reactions or desulfuration reactions brings about the finally the inorganic sulfur. The H2S derived from cysteine, it may be oxidized to first sulfides and then to sulfate. So the H2S formed from cysteine will be finally oxidized to form sulfate. This sulfate is very important in the formation of active sulfate. So this active sulfate, the other name is phosphoadenosine phosphosulfate, that is PAPS. Like active methionine, the SAM, here the active sulfate is PAPS, that is phosphoadenosine phosphosulfate. It is formed from the reaction between sulfate and ATP. So this sulfate group is attached to the ribose 5-phosphate of the ATP and forming the phosphoadenosine phosphosulfate. This PAPS is used for the various sulfuration reactions, for example, synthesis of sulfatides and glycosaminoglycans. See, the ATP, when it combines with sulfate, attaches with the sulfate, it forms the PAPS, that is phosphoadenosine phosphosulfate. See, this sulfate is attached to the ribose 5-phosphate of ATP. This is the nitrogenous base adenine present in the ATP, adeno triphosphate. Along with that, the sulfate group is attached, forming the phosphoadenosine phosphosulfate, phosphoadenosine phosphosulfate, which is the active sulfate. Now we'll come to the end of the sulfur-containing amino acids, and this is very important sulfur-containing amino acids. And the next class we'll see the inborn errors of sulfur-containing amino acid metabolism of sulfur-containing amino acids. And now we'll come to the end of this class. Thank you.